Hey, my name is Callie Chappelle, and thanks for tuning into this video. I am a bio student at the University of Michigan, and this video is about chi-square analysis. It is made for the class Bio 305, which is our Introduction to Genetics course. Due to the breadth of the course, I wanted to make a series of videos that highlights the most important aspects of the class, or the things that you really need to make sure you know for an exam. I like to call this quick and dirty because I'm gearing these videos towards students who either would like additional resources to better understand material covered in class, or perhaps, hopefully you're not this person, weren't in class and need to make sure that you're up to speed on what we covered. So, the quick and dirty for this video is literally one sample question. So I just wanted to take a question that was actually on my final exam, that was winter 2014, and go through it step by step, because chi-square, the first time, is a little bit confusing, but once you get it down, it's super easy. But you have to know what to do. So, chi-square, is that a green tea? Maybe this is a question that you're asking yourself right now. No, chi-square is not a green tea. This meme was probably talking about chai, which is also not a green tea. It is a cool statistical method that we use to determine if there is a significant difference between observed values, or experimental results, and expected values, hypothesized results. So why would this ever be helpful in the real world? Well, when you are doing an experiment, you make a hypothesis. And when you complete the experiment, you have results. So you want to know, do these results really fit with my hypothesis? Well, there's an easy way to do it. Use chi-square. So I'm not going to go into the detail of the statistical background for chi-square, but you should know that when you create a hypothesis, or the hypothesized results, we call that the null hypothesis. And that is, in this case, that the observed values, the values that we actually collect down the field, equal the expected. And we're going to look and see whether or not that's true or not based on the table of observed values that we're given. So when you're doing a chi-square question, you'll be given two critical pieces of information, sometimes more. The first is a table of observed values. So this will look like data collected from an experiment. So it'll be like, okay, we crossed 6,000 flies, and 10 of the flies had red eyes, 12 of the flies had white eyes, some of them had yellow bodies, some of them had brown bodies, etc. You'll also be given a critical value table for determining the p-value. That doesn't mean anything to you right now, just know that you'll have it. Then, what do you have to do? Well, you'll have to calculate the chi-square value. That's this little x, which is called the chi-square. Second, calculate the expected values, which may or may not be given. And then third, you need to interpret what it means. So here's a cool backstory about chi. You're probably wondering, if you're like me and grew up in like whiter than white Traverse City, why Christmas is abbreviated Xmas. Like chi, maybe you know this because of random frats nearby where you live, like me last year. Chi is actually like a Greek letter, right? So this is chi. And that's just like usually written when you have like the squiggly X, that's like fancy. And so when they were trying to shorten the word for Christmas, they just used chi. So this is actually chi-mas, Christmas, get it? So that's what chi is. Alright, so here's the question. A petunia plant that is true breeding for a mutant pink flower, blah, blah, blah. I'm not going to read this. Fun story, so trichomes are those little hairs on plant leaves and stems. I was really excited to see this on my exam because I actually work in a plant biolab and we count a lot of trichomes, so I was like super excited for that. But here's the takeaway. So here are the two questions. What's the most likely hypothesis for the test? And then to perform the test, what I want you to do is try to do this on your own. Try to do it. Do it. Pause this video. Do it on your own. I'm going to do it with you. But after you do it, then as I go through how I think about it, you could identify your method of thinking about it, choose to use or not use mine, and then ultimately design the best way for you, because that's really the takeaway. So what is the most likely hypothesis of this test? Well, first, I like to see about what I can eliminate. So we can eliminate A. So A says the plant being tested is a monohybrid with independently assorting genes. Well, we're talking about two different traits, so it's not a monohybrid cross. How do I know that? Well, it says in the question, hang on, let me pull up a different pen color because this is too light for me. I kind of like this orange. Okay, it says we're talking about a mutant pink flower color. It's crossed with the absence of trichomes. So these are two different traits, so it can't be a monohybrid, so that's wrong. All right, the second question is, we can eliminate D. Okay, this is a test cross, and thus a 1-to-1 to 1-to-1 ratio is expected in the progeny. Well, that's not always true, because you can have a test cross without having a 1-to-1 to 1 ratio. Also, this is not a test cross, which is where you cross with a homozygous recessive. It says here that F1 was selfed. Well, selfing is not a test cross unless you are completely homozygous recessive, which appears it's not, which means that this is wrong. Okay, that brings us to determining whether or not it's B or C. So the fundamental question, the fundamental distinction between B and C is, do all progeny have equal viability? And do they have to have equal viability to make a Mendelian assumption to predict the outcome? Well, the answer is yes, because think about it. If randomly all plants with pink flowers, all plants with a certain genotype and no trichomes died, how could we expect that? We have no way of determining our expected values, which are not given in this table, which means that we have to assume that all progeny have equal viability, even to create a set of numbers that we can even run the chi-square with, which means the answer is C. The plant being tested is a dihybrid, which we know because we have them two different traits, with independently assorting genes. Okay, we have to have independently assorting genes to fulfill Mendelian ratios, and all progeny are of equal viability. That's necessary for determining the outcome of the question. All right, so we need to perform the actual chi-square test. So first, we need to figure out the cross. Second, we need to calculate expected values. Third, we need to calculate the chi-square value. Fourth, we're going to find the p-value. And fifth, we're going to be interpreting it. So the first thing that we need to do is isolate the most important parts of the question. So here I've created this handy-dandy table. So here in this column is what the question says. Here is real talk, so figuring out like what the hell that even means. And then third, I'm actually drawing the cross. So this is where I'm transforming this into understanding what it's saying, and this is where I'm actually transforming it into symbols that we use in genetics to figure out the answer. Okay, so the first thing the question says, we have a petunia plant that is true breeding for a mutant pink flower color that's crossed with a petunia plant that is true breeding for the absence of trichomes. So what does this mean? Well, it means that we have two things that are the same. So the petunia, the pink color I did in pink, and then the trichomes I did in green. So this means that if it's true breeding for pink, we're going to have traits related to pink here in pink, and then that it's true breeding, that means that it has to be the same. So true breeding means that through multiple generations, you're not getting a mix of genotypes, which means that it has to be homozygous. So true breeding, we should write that down. True breeding equals homozygous. And why is that? Because, like, let's say we did a cross, of heterozygotes. Well, heterozygotes produce many phenotypes, not just parental. So let's say that the A is dominant and the A is recessive. This is going to be a different phenotype than these other ones, which means that it will not be true breeding, which means that all true breeding are homozygous. Okay, the next part of the question says all F1 plants from this cross exhibit wild-type purple flowers in the presence of trichomes, which means this, right? So the mutant pink color is recessive because it doesn't show up when crossed to a true breeding wild type, and the absence of trichomes is also recessive because when you cross these two, you get a different phenotype, okay? So we know that this has to be the result, okay? Because this gives us information that pink is recessive and that trichomes is recessive, which means that when we started out reading the question, all we knew is this. Now, because of this information, we can fill it in here, okay? So this is a different phenotype than this because this has dominant, right? And this is recessive. This is a different phenotype than this because this is recessive and this is dominant. The only way to produce this is, and we know these are true breeding, is if these are the parental phenotypes. Did you see where that came from? If not, think about it a little bit more or gain extra help. The last part of the question is telling us that the F1 was self. So selfing means that you just cross it to yourself or functionally another sibling that has the exact same genotype as you. So we know that the F1 cross looks like this. Honestly, this is the most challenging part of the question, is to actually figure out what the hell it's talking about. Okay. Next thing, we're going to make a table to compare the observed and the expected, okay? So here's the phenotype. This was given in the question, or this was like determined from the question. Here's the genotype of the F1. So we're looking at the F1 generation. Here's the genotype of all the progeny, and here are the observed. One thing that I should have done was write, again, what the F1 cross was. So this was on the previous slide, but just a reminder, the F1 cross was big P, big T, little t, cross big P, big T, little t. These two are the same because it said they were self. Alright, so here are the possible genotypes of all the progeny. Big P something, little t something, big P something, little t, etc. I can correlate this with the phenotype because we know that purple is dominant, we know that trichomes is dominant, we know that no trichomes is recessive, and we know that pink is also recessive. So that's how I correlated these genotypes with these phenotypes. Alright, next, I made a table for observed, okay? So these were given in the problem. So I didn't copy this in the previous slide, but it said like purple trichomes this, purple no trichomes that, blah, 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 etc. Now, the part that we need to calculate is this expected table. So we'll remember the whole purpose of a chi-square is to compare the observed to the expected. And we don't know what the expected are. That's why we had to figure out what the cross is, so we can use our Mendelian ratios and probability to figure out what expected should occur. Now, again, right down here, here's the F1. How do we even figure out what the expected is? Well, we're going to figure out expected by separating the two genes because of the law, Mendel's law of independent assortment means that these two genes, the P gene and the T gene, will segregate independently. That means that we can calculate the probabilities of particular outcomes with P and T completely separately. So, in this context, we're going to do each of the P crosses separate from the T crosses, and then, because they are independent, remember the probability lecture, we can multiply. Independent means multiply. So let's do the crosses, right? So we cross big P to little p, and we get 3 big P's and 1 little p. So that means the probability of showing the dominant phenotype, which remember is purple, is 3 out of 4, and the probability of getting the recessive phenotype, which we know is pink, based on other parts of the question, is 1/4. All right, let's do the same thing with the heterozygote for T. So we get the same thing. So we have 3/4 likely will show the dominant phenotype of trichomes. Oops, I can't spell. Yeah, this is like my eternal problem. And 1/4, no trichomes, no little hairs. Really sad. I'm gonna make a sad face here because that makes me sad because trichomes are pretty cool. Alright, so now we're gonna look at this phenotype and this genotype and figure out what is the expected probability. Well, what is the probability of having P, the dominant phenotype, purple, and trichomes, the dominant phenotype, trichomes? Well, because these are independent, we're going to multiply. So we're going to multiply probability of P, 3/4, times the probability of big T, 3/4. Then we're going to multiply that by 180, because that's the total, to get our expected number of individuals with that phenotype if we have independent assortment and, Mendelian ratios. So that comes out to 495. So we did the same thing with purple no trichomes. Probability of being purple, aka the probability of having this genotype, times the probability of having this genotype. So that's 3/4, right, because that's down here. And then the probability of having this genotype, little t, is 1/4. And then you get 165. Cool, so we did that with all of these, which I'm going to skip over for purposes of time. And we're going to go to the next step. So So the next step is to calculate the chi-square value for each phenotypic category and then calculate the sum. Well, that sounds all good and dandy and really academic. What does that mean? Well, the chi-square value is calculated, each chi-square value for each of these phenotypes is calculated utilizing this equation. Now, the total chi-square equals the sum of the individual chi-squares, okay? So we're going to do this one by one. So here's the observed. That's just given in the problem. So we don't have to worry about that. But here's the expected. So purple and trichomes. Let's just do this once. So here's purple trichome. So we're going to do observed, so that's 494 minus expected 495 squared over expected 495. And that comes out to 0.00202. Okay, so that is what went up here in this aspect of the table. So I went through and did that for all of these, and then I summed up all of these, so oops, it deleted everything. Cool, that gives me more space to do this up here. 0.00202 plus 0.0545 plus 0.006061 plus 0.01818 comes out to 0.8080808. Wow, that's a cool number. And that is our chi-square value. So remember this, remember how we got it, because the next step is to find the p-value. So the p-value is the probability, that we can accept the null hypothesis. Now that's not exactly like an actual statistical definition of the p-value, but that's what you can think about when you're thinking about these problems. So we're going to find the p-value given the table. So here's a table. Remember back when I was saying the things that you'll be given. This is something that you'll be given. You'll never have to memorize this. And we have to find our chi-square value. So remember our chi-square value, our chi-square total was 0.08 something, right? Was it 0.08? Let's look back. Yeah, it was 0.08, 0.081. So we're going to have to find that value in this table. So first, how do we read this table? Well, here are degrees of freedom. You don't need to worry yourself about what degrees of freedom actually mean, but they are calculated by number of categories minus 1. Here, this green are the chi-square values, and then here are the p-values, and this is what we're looking for. So degrees of freedom can be calculated as the number of categories minus 1. So the number of categories in this case was, I don't know, let's go back and look. Let's go back and look at the question. So, petunia plant that's true breeding for a mutant flower, all F1. Okay, so here are the number of categories. Purple with trichomes, purple no trichomes, pink with trichomes, pink no trichomes. So there are 4 main categories here. So, degrees of freedom. So, in this case, the degrees of freedom equals 4 minus 1 equals 3. Okay. So first you're going to go to the degrees of freedom category, 3. Then we're going to go over and see where our chi-square is. Now wait a second, chi-square isn't on this list. It's super tiny. It's like 0.081. Whoa, so like if this was a distribution, and this is chi-square, here's like 6.25. We're talking way down here, 0.081. That means, sorry, this probably shouldn't cross the x-axis. It goes like that, actually. That means that we're going to go up to the p-value. Our p-value is going to be way larger, like much, much larger than 0.1. P is huge. So, find the p-value. Oops, where did my mouse go? P is much, much, much larger than 0.10. Okay. Now, let's say that our chi-square value was 7.82. Well, and our degrees of freedom was also 3. Well, we go to 7.82, we'd go up, we'd see our p-value is 0.05. Okay, so that's like how you read this table. And the last thing is to interpret the p-value. So you can remember this easy phrase, if the p is low, the null must go. Okay, so if p is low, if p is much smaller than alpha, and we use alpha, which is our confidence level, as 0.05, then we'll reject the null. Remember, the null is that observed equals expected, and we'll conclude that there is a significant difference between observed and expected. That's that observed does not equal expected at a statistically significant level. Now, if p is much greater than alpha, that's the case here. Remember, p is way greater than 0.10, and we know that 0.10 is greater than 0.05. Then we're going to fail to reject the null, which means and we conclude there is no significant difference between observed and expected. So in normal people speak, we're going to say that observed functionally equals expected. So what does that mean in the context of this testing trichomes and colors? Well, that means that when we actually did the cross, it was what we thought it would be based on Mendelian ratios. So that means that our cross actually was real, like we didn't have any weird confounding factors, because the number of flowers that we thought we should get that fell into each of the categories, purple trichomes, purple no trichomes, pink trichomes, pink no trichomes, was about what we'd expect based on the cross that we performed. So that is a conclusion. So hopefully this video has helped you better understand how to do a chi-square test, and hopefully this Courage baby, or I don't even know what the name's called. This young child is you. That you can do statistics in pen and get the right answer every time. Cool. Take care, and hopefully I'll see you in the next video. Bye.